Good evening to all of you. My voice is audible. Please let me know. Are you able to listen to my voice? Are you able to listen to me? Please let me know so that we will start our class. Please let me know. Are you able to listen to me? Good evening to all of you. My voice is audible. Yeah. Okay. Good afternoon, Anamika. How are you? Everything is fine? Please let me know. Are you able to get my voice? Are you able to listen to my voice? Okay, okay. So just waiting for 1 or 2 minutes more. Otherwise, I will start the session. Today I have to complete two metabolism, which is carbohydrate metabolism and fatty acid metabolism. So these two metabolisms are left. Otherwise, your syllabus is complete. So I am waiting for 1 or 2 minutes more, and then I will start the class. If you have any question or doubt, you can ask. You can ask if you have any queries. So let's waiting for 1 or 2 minutes more, otherwise we will start the lecture. So I will complete the yesterday's homework. Anamika, please let me know. I will complete the homework. So yes, now we will start our today's lecture series. I am waiting for 1 minute or 2 minutes more so that other students will join with us. Otherwise, I will complete the metabolism part. Today is our last lecture. Today is the last lecture series of this series. Today is our last lecture. So I am just waiting for a few more students to join. Otherwise, I will start the session. So we have to complete the metabolism part only, which is left. So we have to complete that, only that part only. in the office that let me share with you the syllabus which we have. Okay. Yes. Good evening, Ashutosh. How are you? So yes, I am going to share the syllabus with all of you. That then Diksha, good afternoon, Diksha. Then after it, this is our last lecture series. This is our last day on this lecture series. We are left only with the metabolism part. Otherwise, everything is covered. So I am sharing the syllabus with you. I will try to complete all things which are there in the syllabus. If anything is left, I will share the PPT or I will try to complete it also. So yes, this is the syllabus which we have. We have covered nucleic acid, DNA, RNA. We have to cover hormonal disturbances. We will try to complete it today. We have covered protein and steroid hormones. Then this LFT and jaundice is also there. We have covered enzymes. We have covered lipids. Our main thing is this metabolism which is left. We have left with fatty acid metabolism and carbohydrate metabolism. So I am now just opening the PPT as we have to complete the part. Yeah. So we have to complete carbohydrate metabolism, which is very important. This part is left. Along with it, we are left with hormonal disorders, jaundice, and just wait, LFT, and yeah, metabolism of fat. So only these things are left, and I will complete it now. Otherwise, we have completed everything. Okay. So I will start with the carbohydrate metabolism. I think we have completed protein metabolism yesterday. Let me check it. Yes. So we have covered protein metabolism yesterday. So today we will move with the carbohydrate metabolism. So I will start with carbohydrate metabolism. So is it visible to all of you? Yes, it's visible. My voice is audible to all of you? Please let me know my voice is audible. My voice is audible to all? Please let me know. So I am going to start with carbohydrate metabolism. My voice is audible. Dikshika, my voice is audible. Diksha, my voice is audible to you. Okay. Thank you, Pooja. So now let's continue. So we have to start with carbohydrate metabolism. Now what is basically carbohydrates? You all know it that carbohydrates are polyhydroxyaldehydes or ketones. They have some nutritive value. Carbohydrates is a classification of carbohydrates, some function of carbohydrates, and metabolism is also related with these carbohydrates. Then we have polysaccharides, carbohydrates. There are some hormones that are also there which are helpful in the carbohydrate metabolism. Some dental aspects are also there. So many things are there. We will start it. So what is basically nutrition? Nutrition is defined as the science of how the body utilizes food to meet requirements for developmental growth, repair and maintenance. And for this we need carbs, we need fats, we need proteins, vitamins, minerals as well as water. For this is our daily intake, the nutrient intake which is needed by the body. So what is basically carbohydrates? They are the most abundant organic molecules on the earth. Carbohydrates are defined as aldehyde or keto derivatives of polyhydric alcohols. For example, glycerol, an oxidation is converted to diglyceraldehyde which is a carbohydrate derived from a trihydric alcohol. So all carbohydrates have the general formula of CnH2 and ON. So this is a classification of the carbohydrates. Can be monosaccharides, disaccharides, polysaccharides, oligosaccharides. You all know about the basic terms which are related to carbohydrates. So carbohydrate is the main source of the energy in the body. Energy production from the carbohydrates will be 4 calories per gram. Storage form of energy is also, so they are stored in the form of starch and glycogen. Excess carbohydrate is converted to fat. We all know that when you are consuming excess carbs, it will be stored in your body. So glycoproteins and glycolipids are the components of cell membranes and structures. Structural basis of many organisms, for example, cellulose of plants, exoskeleton of insects, etc. So this is a biological, some importance of glucose, that glucose is a major carbohydrate, number one. It is a major fuel of tissues, number two. It is converted into other carbohydrates also. So glycogen is needed for storage, ribose is there in the nitric acid, galactose is present in the milk. So all these are the carbohydrates. Okay, they are also present in the lipoproteins, they are present in the plasma membrane. They are also a major integral membrane glycoprotein in case of RBCs. So these are some important functions of carbohydrates. Now what's the biological significance of these carbohydrates? So these are the major source of energy for living organisms. Number one, supplying a huge array of metabolic intermediates for biosynthetic reactions, the structural elements in cell coat or connective tissue. So these are the biological significance of these carbohydrates, that it is the main source of energy. It is the main source of energy for all living organisms, supplying a huge array of metabolic intermediates for biosynthetic reactions. So digestion. So partially digested in mouth by the salivary amylase. It is about the carbohydrates. Like proteins is not digested in the mouth, but yes, these carbs are digested in the mouth, while salivary amylase in stomach there is no digestion takes place. Complete digestion and absorption will be takes place in the small intestine only. So this is the metabolism, that thousands of chemical reactions are taking place inside the cell in an organized, well coordinated and purposeful manner. All these reactions are known as metabolism. The type of metabolic pathway, we have catabolic pathway, we have anabolic pathway, we have amphibolic pathway. So this is the food molecules. The simpler molecules will get changes into amphibolic pathway, which will either break down into CO2 and water, or they may be used as further used as proteins, carbohydrates, lipids, or nucleic acids. So now the main thing which we have to understand is the major pathways of carbohydrate metabolism. So we have glycolysis, we have PCA cycle, glycogenesis, glycogenolysis, HMP pathway, uronic acid pathway, galactose metabolism, fructose metabolism, amino sugar metabolism. So all these metabolisms are there. So the metabolism of glucose, it needs aerobic oxidation. Glycolysis is there, glyconeogenesis, pentose phosphate pathway, glycogenesis, glycogenolysis, uronic acid pathway. So from the glucose we are able to get glycogen, we are able to get ribose, we are able to get lactate, amino acid, glycerol, starch. So many things are there which are related with the glucose. So either we will have to do glycogenesis or glycogenolysis, glycolysis, glyconeogenesis, pentose phosphate pathway. So there are many pathways which are related with the carbohydrate metabolism. Now this is the cell organelles, this is the cell and the organelles which are there present inside the cell. So after it we have animal cell. So this is the nucleus. In the nucleus we have plasmic reticulum is there, mitochondria is there, Golgi complex is there. So this is the organelles which are present inside the cell. Now we have to discuss glycolysis. Now what is glycolysis? The anaerobic catabolic pathway by which a molecule of glucose is broken down into two molecules of lactate. So in the presence of anaerobic condition, glucose will get changed into 2 lactic acid. So all of the enzymes of glycolysis locate in the cytosol. So glucose get changes into lactic acid. So this is glycolysis. This is also known as EMP pathway because of the name of the scientist, which is embedded in mighty whole pathway. So it is basically glycolysis. So glycolysis is defined as a sequence of reactions converting glucose to pyruvate or lactate with the production of ATP. So from glucose, what we will get? We will get pyruvate or lactate with the help of or with the production of the ATP. So glycolysis is defined as a sequence of reactions converting glucose to pyruvate or lactate. So these are the salient features. It takes place in all cells of the body. Enzymes present in the cytosomal fractionation of the cell. Lactate is the end product in the anaerobic condition, and pyruvate is the end product in case of aerobic condition. So we have either lactate or we have either pyruvate from the anaerobic conditions. So after it we have tissue lacking mitochondria are the major pathway of ATP synthesis, very essential for the brain, dependent on the glucose for energy. It is a central metabolic pathway. Reverse of glycolysis results in glyconeogenesis. So from glucose glycolysis, what we get? From glucose we are getting pyruvate, and in case of glyconeogenesis we will get the glucose back. So these are the reactions of glycolysis. Now we have energy investment phase, we have secreting phase, we have energy generation phase. So this is the glucose, the number one is energy investment phase. What will happen now? The glucose is phosphorylated to glucose 6-phosphate by the presence of enzyme hexokinase. So this is your homework also. You have to draw these pathways like glycolysis, all the pathways which will be there, because today is your last lecture, so you will have the time of 2 days to submit your assignment. So from the glucose we will get glucose 6-phosphate by hexokinase. Glucose 6-phosphate undergoes isomerization to give fructose 6-phosphate in the presence of phosphohexose isomerase. Fructose 6-phosphate is phosphorylated to fructose 1,6-phosphate by phosphofructose kinase. This fructose 1,6-phosphate gets changes into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Let me show you the diagram. So from the glucose we will get changes into glucose 6-phosphate in the presence of enzyme hexokinase. This glucose 6-phosphate then changes into fructose 6-phosphate in the presence of enzyme phosphoglucoisomerase. This fructose 6-phosphate then gets changes into fructose 1,6-bisphosphate, which will changes into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. From this we will get 1,3-bisphosphoglycerate. From 1,3-bisphosphoglycerate we will get 3-phosphoglycerate in the presence of enzyme phosphoglycokinase, and from 3-phosphoglycerate we will get 2-phosphoglycerate. From 2-phosphoglycerate we will get phosphoenol pyruvate, and at the end we will get pyruvate from the a single molecule of glucose. What we will get? We will get 2 molecules of pyruvate. So this is how the ATP production is there. There are many steps in which ATP is needed, and in many steps ATP is produced. Like here we are getting the ATP. So here at the end, how many molecules will be there, how many ATPs we are using for this reaction, you have to tell me. So this is a clinical aspect. This is lactic acidosis basically. This is again the condition where there is because of some exercise shock or respiratory diseases, we have this condition in which we have pain and severe shock also. After it we have another thing which is glycolysis, in which glucose gets changes into pyruvate. So if oxygen is not present now, this pyruvate will further changes into lactic acid. So this is the glycolytic pathway in which glucose gets changes into pyruvate. It involves 10 steps. So you have to draw this glycolytic pathway. Okay. After it we have another pathway which is known as, this is again the same pathway which is the very first pathway. This is the glycolysis. That means from glucose what we will get? Pyruvate. And if the oxygen is not there, the pyruvate will get changes into lactate. After it, so in this glycolysis pathway basically how much ATP we are using, you have to tell me. Now there are some enzymes also which are there in the glycolysis. We have hexokinase, we have phosphofructokinase, and we have pyruvate kinase. So these three enzymes are quite important in the case of glycolysis. After which we have another cycle which is there, which is known as, yeah, so now we have another cycle which is the TCA cycle. So if you are able to see the process of complete oxidation of glucose to CO2 and water with liberation of energy as the form of ATP is named as aerobic oxidation, the main pathway of G oxidation. Now we have the process of aerobic oxidation. The very first thing is glucose will get changes into pyruvate. The number one is glucose will get changes into pyruvate in the glycolytic pathway in the cytosol. Now this pyruvate then enters into the mitochondria, and this pyruvate gets changes into acetyl coenzyme A. This acetyl coenzyme A then changes into CO2, water and ATP. So all these things are occurring inside the mitochondria. So now this pyruvate will get changes into acetyl coenzyme A, and this is irreversible reaction. So it is not back reaction, it is irreversible, and from that pyruvate when we get acetyl coenzyme A, this reaction is occurring inside the mitochondria only. So this is pyruvate dehydrogenase complex. The pyruvate dehydrogenase is made up of these enzymes, and even some cofactors are also attached. This is pyruvate dehydrogenase complex. This is the structure of the pyruvate dehydrogenase complex. Where we need this pyruvate dehydrogenase complex? Here. When pyruvate gets changes into acetyl coenzyme A, at that time we are needing the pyruvate dehydrogenase complex. So this is E1, E2, E3, TPP, FAD. This is pyruvate dehydrogenase complex. Now what happened now? When pyruvate gets changes into acetyl coenzyme A, now what will happen now? Now there is another cycle which is the TCA cycle, also known as Krebs cycle, citric acid cycle, tricarboxylic acid cycle. This is the second step after glycolysis, so you have to draw this cycle also. So here what happens, it essentially involves the oxidation of acetyl coenzyme A to CO2 and water. So this cycle utilizes about 2/3 of total oxygen consumed by the body. So this cycle utilizes 2/3 of total oxygen utilized or consumed by the body. So this is a brief history of this is the scientist whose name is Hans Krebs in 1937. He was able to study this, and the location is in the mitochondrial matrix. So from this acetyl coenzyme A, what we will get? We will get CO2 and water. So TCA used name because at the outside of the cycle tricarboxylic acids participate. So let us see this now. So this is the next cycle, which is TCA cycle. From pyruvate we will get acetyl coenzyme A. From this acetyl coenzyme A we will get citrate. From citrate we will get isocitrate. This isocitrate then changes into alpha ketoglutarate. From this alpha ketoglutarate we will get succinyl 4-enzyme A. Succinyl 4-enzyme A gets changes into succinate, which further changes into fumarate. Fumarate changes into malate, and at the end we will get oxaloacetate. So this is the whole reactions which are occurring in the TCA cycle. Again there we are using the ATPs also. So TCA cycle is strictly aerobic in contrast to glycolysis. So here we are using the 12 ATPs from one acetyl coenzyme A. So here we are able to get 12 ATP molecules. So during the process of oxidation, acetyl coenzyme A via citric acid cycle, oxidation of 3 NADH by electron transport chain. This is substrate level phosphorylation. So this is the aspects of TCA cycle. Mitochondrial encephalopathy occurs due to fumarase deficiency. Mitochondrial myopathy affecting both skeletal muscles and brain. So you have to draw this TCA cycle also. After it we have another term which is known as glyconeogenesis. Now what is glyconeogenesis? It is a synthesis of glucose from non-carbohydrate compounds. Like there are some compounds like lactate, pyruvate, amino acids, from that compounds we are getting the glucose. So it is a synthesis of glucose. It is glyconeogenesis. Genesis means synthesis. Glyco means glucose. So synthesis of glucose, but from non-carbohydrate compounds. So major substrate precursors are lactate, pyruvate, glycogenic amino acids, propionate and glycerol. It takes place in the liver, kidney matrix, occurs in the cytosol, and some produced in the mitochondria also. So this is the importance of glyconeogenesis. It occurs in the brain, in the CNS, erythrocytes, testes, kidney, medulla depend on the glucose. And under aerobic condition, glucose is the only source to supply skeletal muscles. So occurs to meet the basic requirement of the body for glucose in fasting for even more than a day. So when you are fasting, at that time you are using this glyconeogenesis. It effectively clears certain metabolites produced in the tissue that accumulates in the blood. So this is a reaction of glyconeogenesis, that is from you can say from succinate, from fumarate, from oxaloacetate. What we will get the glucose. These are the pathways only. You have to know the pathways only. Now we have glycogen metabolism. What is glycogen basically? So glycogen is a storage form of a glucose in animals, stored mostly in liver and in muscles. Due to muscle mass, the quantity of glycogen in muscle is 250 gram and in liver it is 75 gram. So it is stored as a granules in the cytosol. The function is liver glycogen is here to maintain the blood glucose level. So this is glycogenesis now. Now what is glycogenesis? It's a synthesis of glycogen from the glucose. When the glucose is present in excess amount, then we have to synthesize it. We have to change it into the glycogen. After it, we have the reaction which takes place. So the reaction will take place in the cytosol. Basically, it requires UTP and ATP along with glucose. So this is the process. From glucose we will get like glucose 6 phosphate. From glucose 6 phosphate it will changes into glucose 1 phosphate. From it we will get UTP glucose. UTP glucose that changes into glycosyl units. It will later changes into glycogen. So this is the storage of glucose in the form of glycogen, and this is known as glycogenesis. So this is the third pathway which we have to draw. After this we have glycogenolysis. Now here is the breakdown. So it's the degradation of the stored glycogen in liver and muscle constituents glycogenolysis. It is a irreversible pathway. What happens in the glycogenolysis? Glycogen gets breakdown into glucose. So glycogen gets breakdown into glucose. So these are some diseases which are associated with glycogen. We have Dyke's disease, Pom's disease. These are maybe because of the deficiency of the enzymes. We have some diseases which are there. Like this is von Dyke's disease, this is Pom's disease. Similarly, we have Cory's disease, also known as Fodby disease. Then another thing is hexose monophosphate shunt. Now what is HMP pathway? What is HMP pathway? Are you getting my voice? My voice is audible to you. Please let me know. Diksha, my voice is audible. Please let me know. Yes, please let me know my voice is audible. Diksha, are you getting me? Please reply, dear. Are you getting me? Then I will continue. Diksha, are you getting my voice? Okay. Thank you, Surabhi. So now we have another pathway which is known as the HMP pathway. So any one of you please write down the homework alongside also. So now in the meantime we have to draw three life cycles. We have not three, four. Glycolysis, then PCA cycle, then glycogenesis, then glycogenolysis. All the pathways which we are discussing right now, you have to draw the diagrammatic representation of it only. Okay. So please, anyone please write the homework anywhere. So after it we have the another pathway which is known as SMP pathway. So we are going with the SMP pathway. So now what is basically SMP pathway? This is also known as hexose monophosphate shunt or PP pathway or pentose phosphate pathway, or also known as phosphoglyconate pathway. So this is an alternative pathway to glycolysis and PCA cycle for the oxidation of glucose. So basically this cycle is for the oxidation of the glucose to anabolic in nature, since it is concerned with the biosynthesis of NADPH and pentoses. So unique multifunctional pathways, the enzymes are located in the cytosol. Tissues active: liver, adipose tissue, adrenal gland, erythrocytes, testes, and other lactating mammary gland. This is the area where we have this PP pathway. So this is the alternative pathway of the oxidation of the glucose molecule. So this is SMP pathway. We have glucose. From glucose we are getting glucose 6-phosphate. Now this glucose 6-phosphate get changes into ribulose 5-phosphate. That is my point. So glucose 6-phosphate get changes into ribulose 5-phosphate. This will get changes into ribose 5-phosphate, cellulose 5-phosphate, which again changes into cerohexulose 7-phosphate, and then again it gets changes into fructose 6-phosphate and isolose 4-phosphate. Similarly, there we will get fructose 6-phosphate. So, what is the significance of this attention? So, control for its derivatives are useful for the synthesis of nucleic acids and nucleotides. NADPH is required for the reductive biosynthesis of fatty acids and steroids, for the synthesis of certain amino acids, antioxidant reaction, hydroxylation reaction, detoxification of the drugs, phagocytosis, preserve the integrity of the RBC membrane. For that only we are using the NADPH. So this is the clinical aspect of it. The glucose 6-phosphate dehydrogenase deficiency. If we have the deficiency of glucose 6-phosphate dehydrogenase, what will happen then? It will lead to a heightened sex linked state. Red blood cells will be there. Impaired synthesis of NADPH, resistance towards malaria. So these are some aspects of this SMP pathway. But you have to remember this pathway only, that from 6-carbon glucose, what we will get? We will either get 4-carbon compound, or we will get 5-carbon compound, and we will get 3-carbon compound. So this is the thing which is there in case of HMP pathway. Then we have another pathway which is known as uronic acid pathway or glycuronic acid pathway. What happens in this? This is the alternative oxidative pathway for glucose. Synthesis of glycuronic acid, pentoses and vitamin. Normal carbohydrate metabolism. Phosphate esters are involved. But in uronic acid pathway, free sugars and sugar acids are involved. So these are the steps of reaction which occurring: formation of QTP glycuronate, conversion of GGP glycuronate to L-gluconate, synthesis of ascorbic acid from animals, oxidation of L-gluconate. So this is the D-glucuronic acid. It gets changes into L-gluconate and it gets changes into pentose phosphate pathway. This L-gluconolactone then gets changes into 2-gluconolactone, it gets changes into L-ascorbic acid. So this is the thing which is there in case of SMP pathway. Then we have another pathway which is known as Now these are the physical aspects of it. They have the effects of drugs also. They are including the pathway to achieve low synthesis of glycoronate from glucose, barbitol and chlorobutanol. And then we have some another diseases which are there because of the deficiency only. This is the metabolism of lactose. From lactose we will get glycolysis. From lactose we will get lactose 1-phosphate. From lactose 1-phosphate we will get glucose 1-phosphate. And from glucose 1-phosphate we will get glucose 6-phosphate, which gets changes into glycolysis. Similarly, we have metabolism of fructose also, metabolism of amino sugars also, which are there. Now, another thing which is there is the electron transport chain reaction. Now, what is basically electron transport chain? So, electron transport chain is a series of protein complexes located in the inner membrane of the mitochondria. So, we have some complexes like complex 1, complex 2, complex 3, 4 and 5, which are located in the inner membrane of the mitochondria. All these are helpful in the production of the ATP. Now there are some other glycosaminoglycans which are there, like we have monosaccharides, the deficiencies of which causes Hunter syndrome. Then we have some hormones also which are quite helpful in the metabolic pathways, like we have let me show you some hormones. Like there is the glucagon, the hormone which is there. Then we have thyroid hormones are there, which stimulates glycogenolysis as well as glyconeogenesis. Then we have glycocorticoids also. Epinephrine is there, which is secreted by the adrenal medulla. It stimulates glycogenolysis in the liver and the muscles. It diminishes the release of insulin from the pancreas. Some growth hormones are also involved here. Sex hormones are also there. Now these are some diseases which are related with carbohydrate metabolism. The very first is diabetes mellitus. You all aware of it. Then there we have some another aspects of these carbohydrates. Let me show you. Like this is cystic fibrosis. This is a disease which is related with the metabolism part. Basically, it leads to gout also. These are the foods from where we will get the carbohydrates. So carbohydrates are the major source of energy for living cells. Glucose is the main central molecule in the carbohydrate metabolism. So we have TCA cycle, we have citrate cycle or TCA cycle, we have Krebs cycle, we have one more cycle which is there, which is the ETC, energy transport chain is there. So you have to draw. We have pentose phosphate pathway cycle is there. We discussed this PPP pathway already. And then we have glycogen synthesis and catabolism which is there. So glycogen synthesis is known as glycogenesis. So we have glycogenesis, we have glycogenolysis. So we have glyconeogenesis also. It is a process of transformation of non-carbohydrates to glucose or glycogen. The material is lactate, glycerol, pyruvate and glycogenic amino acids. So you can please go through all these cycles as you have to draw all these pathways like glyconeogenesis. So you have to draw all these cycles. This is your homework from this carbohydrate metabolism pathway. So is it clear to all of you? Please let me know. Is it clear to all of you? Please let me know so that we will move further. So after it we have the next cycle, which is metabolism of fat. But before it, I think we will go with the hormonal disorders which are there in your syllabus. So metabolism part, before it, I will start with hormonal disorders. Hormonal disorders are also in your syllabus, the disorders which are related with the hormones. So I will try to complete it first. So this is the hormonal disorders. Yeah. These are the disorders which are because of these are the disorders of pituitary gland, disorders of thyroid gland, adrenal gland, and islets of Langerhans, disorders of ovary, testes, all these. So what is basically endocrine disorders? They may be subdivided into three groups. So endocrine gland hyposecretion. Now there are three types of hormone secretion. When hyposecretion is which leading to the hormone deficiency. Endocrine hypersecretion leading to a hormone excess when the level of hormone is high. Then we have tumors of endocrine glands. So we have three types. We have hyposecretion, we have hyper, and we have tumors also. Tumors benign or malignant of endocrine glands. So diagnosis of endocrine disease may be difficult. It is often not possible to directly assess hormone levels in the blood, making indirect measurements necessary. So these are the hormones which we have studied on the very first day. We have hypophysis, thyroid, pancreas, testes, ovaries, adrenals. There are some deficiencies which are there. Now number one is hypophysis. Hypothalamus, you all know, known as a master gland, as the hormones secreted are very vital for proper development and functioning of the body. Disorders are classified under hyper and hyposecretion of the hormones and pituitary cancer leading to the alteration of the structure of the hormone. So the major disorders of pituitary are acromegaly, diabetes insipidus, hypopituitarism, and pituitary tumor. So this is the diseases which are related with it. This is acromegaly. Now what is acromegaly? It is caused by excessive secretion of growth hormone. So the growth hormone is more excessively secreted. It leads to swelling of hands, legs, and soft tissues. What are the symptoms? Swelling of hands and feet. Facial features become porous as bones grow. Protruding jaw, degenerative arthritis, enlargement of the organs. So this is acromegaly when we are having the excess amount of the hormone. Diabetes insipidus is caused due to insufficient production of antidiuretic hormone ADH. So it is characterized by excessive thirst and excretion. So again and again there is a thirst. That means need to drink water and excretion of large amount of urine. So symptoms are extreme thirst, blurred vision, dehydration, extreme urination, fever, vomiting, and diarrhea. So these are the cases in case of diabetes insipidus. Okay. So after it we have hypopituitarism. Now what is hypo? It is basically the deficiency of all hormones, anterior pituitary hormones, which is more common than the individual hormone. So we have luteinizing hormone, we have follicle stimulating hormone. What happens here? So women experience infrequent menstrual periods and infertility. Women experience infrequent menstrual periods and infertility. Men lose facial and trunk hair as well as suffering decreased muscle mass and anemia, increased risk of osteoporosis. So this is the case in case of luteinizing hormone. So similarly we have growth hormone. If there is a deficiency of growth hormone, what will happen? Decrease in muscle mass, central obesity, impaired attention and memory. Children experience growth retardation and short stature. So basically, if there is a deficiency of growth hormone, the growth will be less. Similarly, we have adrenocorticotropic hormone, in which we have lack of production of glycocorticoids such as cortisol. Fatigue is there, weight loss, delayed puberty. All these are the situations which are because of this adrenocorticotropic hormone. Similarly, we have thyroid stimulating hormone. It will lead to hyperthyroidism. What happens here? Tiredness, intolerance to cold, constipation, weight gain, hair loss, low blood pressure. So all these are the symptoms. Like if prolactin is there, it plays a very important role in breastfeeding. or inability to breastfeed may point at abnormal low prolactin levels. Antidiuretic hormone, inability to concentrate the urine, extreme thirst. Oxytocin deficiency generally causes few symptoms as it is only required at the time of childbirth and breastfeeding. Now we have some hormones like thyroid hormones. Some diseases are there which are related with the thyroid hormone. The number one is keratinism. What happens in keratinism? It is a condition of stunted physical and mental growth. Patient is dwarf with severe mental defect, coarse dry skin, deficient hair and teeth, retarded skeletal growth, reduced BMR. So this is keratinism. Then we have maxedema. So this is the situation in case of maxedema. It is caused by diminished production of thyroxine. Condition is called maxedema as gelatinous mixture of mucoprotein and extracellular fluid is deposited in the intracellular space, specifically specifically in the dermal connective tissue, giving it some another type of appearance basically. So, rate of metabolism in all tissues is decreased to half. So, this is maxedema. After it, we have Hashimoto thyroiditis. What is happened in Hashimoto? It is an inflammation of the thyroid gland. It is an autoimmune disorder wherein the antibodies. It is an autoimmune disorder wherein the antibodies attack the thyroid gland. So the symptoms are goiter, fatigue, muscle weakness, and weight gain. So this is Hashimoto thyroid thyroiditis. Then we have Graves' disease. This is the Graves' disease. So what happened in case of Graves' disease? It is an autoimmune disease where the thyroid is overactive, producing an excessive amount of thyroid hormone. This is caused by autoantibodies to the TSH receptor that activate that TSH receptor, thereby stimulating thyroid hormone synthesis and secretion and thyroid growth. So eyes may look enlarged because the eye muscles swell and push the eye forward. Weight loss in spite of increased appetite, increase in BMI. So the size of eyes become quite large. So these are another symptoms. Swelling is there, weight loss is there, muscle wasting is there, tremor is there. So these are related with Graves' disease. Then we have goiter. You all know about goiter, in which we have a swelling in the thyroid gland. So this is goiter. After it we have adrenal gland. Some diseases, some imbalances are there because of adrenal gland. This is Cushing syndrome. What happens in this? Obesity is there, moon face is there, growth of fat pads along the collarbone and on the back of the neck, purple or red streak on the trunk, buttocks, arms, legs, breast, proximal muscle weakness. So this is Cushing syndrome. Then this is Addison disease. It is also a rare endocrine disorder wherein the adrenal glands produce insufficient steroid hormone. What happens in this disease? Low blood sugar, darkening or hyperpigmentation of the skin, muscle weakness, diarrhea, headache and sweating is there. Then after it we have another diseases which are related to pancreas. The very first is diabetes mellitus. Then the another is pancreatitis. What happens here? If you are see, the swelling is there in the specific region. So nausea, vomiting, fever, swelling in the upper abdomen, severe abdominal pain is there. Then some diseases are related to the ovary, like we have amenorrhea, we have hypogonadism, we have polycystic ovary, PCOS. So this is polycystic ovary. Some diseases are related with testes also. Hypogonadism. Okay, testicular cancer is there. So these are all disorders which are related with the hormonal. So these are the disorders which are the hormonal disorders. Getting my point? Are you getting it or not? So shall we continue further? Only one topic is left. Shall we continue it now? Please let me know. Shall we continue it now? Okay, so now we will move forward to our next topic, which is jaundice. Let me start it now. Jaundice, you all are aware of this jaundice, which is, but still I will try to complete it. Let me open it. So what is basically jaundice? It is a yellowish discoloration. It is the yellowish discoloration of the of the tissues due to the blood due to the deposition of bilirubin, which occur in the presence of hyperbilirubinemia. So basically, it is a yellowish discoloration of the tissue. So jaundice, you all maybe know about it. So it is evident when serum bilirubin crosses 3 mg per dl. So jaundice is latent, that is clinically non-evident, unconjugated hyperbilirubinemia when the level of bilirubin is less than 15% of total serum bilirubin. Then we have conjugated hyperbilirubinemia when the level of bilirubin is greater than 15%. So this is jaundice. I think just 1 minute. Just open the this is basically jaundice which I want to discuss. Yeah, jaundice is basically what basically jaundice is. Jaundice can also result from the excessive breakdown of RBCs and too much bilirubin is released into the bloodstream. When too much bilirubin gets released into the bloodstream, at that its situation is known as jaundice. So this is the things we have liver. In the liver we have an enzyme which is bilirubin. When it is just released too much, it leads to jaundice. So jaundice is a yellow discoloration of the skin, mucous membrane and white of the eye caused by the increased amount of bilirubin in the blood. So bilirubin level exceeds up to 3 times than the normal. The normal value should be 0.3 to 1.2 mg. The normal value should be 0.3 to 1.2 mg per dl. There are some types of jaundice. We have hemolytic jaundice, we have hepatocellular jaundice, and we have obstructive jaundice. So we have three types of jaundice which is there. There is yellowish skin, light grey or clay colored stool, itchy skin, nausea, lack of appetite, tea colored urine, prothrombin time increase. So all are the symptoms of jaundice. The eyes become yellowish. So due to the chemical toxin in blood transfusion, increase the destruction of RBCs, increase the amount of unconjugated bilirubin in the blood, and hepatocellular obstructive causes the jaundice. So when the level of bilirubin get increased and action of bilirubin in the urine reduce fetal urobilinogen because it does not reach in intestine, prolonged prothrombin time. All these are the things which are there. And then we have some drugs also, medicines also for this jaundice. Like we have to take high protein diet, high carbohydrate diet, keep glucose water. These are some you can say some preventive measures also related to jaundice. After it we have one more term which is LFT. Now what is LFT? So LFT is the liver function test. When we have to do the we have to know the functioning of the liver, then we have LFT. The liver is the largest organ of the body, weighing about 1.5 kg. It is also known as the kitchen of our body. So in feed state glycogen synthesis and excess glucose is converted to fatty acid and then PEDS, which gets incorporated to VLDL and transported to the adipose tissue. So this is a protein metabolism. What happens here now? Like in case of lipid metabolism, we have to synthesize lipoproteins, phospholipids, cholesterol. In case of fatty acid, we have to synthesize beta oxidation, ketone bodies. So these are the some terms which are important in the liver. So these all things are occurring inside the LFT, or you can say in the liver. There are some special tests which are there in the liver. for detecting some diseases basically, like bilirubin. If you have to know the amount of bilirubin in the body, for all this, jaundice is again here. I'm not going to discuss these tests only. You can read these tests by your own self, because these are only the tests which are there, liver functioning tests which are there. So I think you can go it by your own self. The diseases which are related with it, like we have cirrhosis, alcoholic hepatitis, jaundice. So you can go it by your own self. I will not discuss these tests only. Now the last thing which I have to discuss is the metabolism of fat. So in the metabolism of fat also, this is your homework also, you have to tell me the important pathways which are there in the metabolism of fat. This is the whole, these slides are whole regarding how the fat digestion will start, how the process of digestion will start, the enzymes which are needed in the pancreas, in the mouth, in the gallbladder, in the small intestine, and then we have absorption and transportation also of these fat molecules. Okay, so this is the fat metabolism. How we are able to break this fat molecule, and when we have to break the fat molecules, during when we do not have the glucose, at that time we need the fat metabolism. So there are two terms which is here. We have LDL, we have HDL. You must know about all these terms also. So this is how the digestion of fat occurs in the digestive tract. You can go it by your own self. Then absorption of fats is there. This is how the body stores the fats. Then oxidation of fat is there. Now there is one cycle which is known as beta oxidation of fatty acid. That is your homework. You have to tell me about the beta oxidation of fatty acid. Let me show you the beta oxidation of fatty acid. I think we have it. The yeah. So this is your homework. You have to tell me the beta oxidation of fatty acid, in which we are able to do 4 steps. The first step is dehydrogenation, number 2 is hydration, number 3 is again this dehydrogenation, and last we will get the enzyme. So yeah, so we have dehydrogenation. The number 1 is dehydrogenation, then hydration, then dehydrogenation, then cytolytic cleavage. So what happens in this? First of all, acyl coenzyme A from the cytoplasm enters into the mitochondria. Then this coenzyme A gets changes into, yeah. So this coenzyme A uh, gets changes into anyl coenzyme A. So if you are able to get here, we are having the double bonds. The hydrogenation of the fatty acyl coenzyme A to make trans double bond between alpha and beta carbon. Hydration of the double bond. After it, we will get the from this palmitoyl coenzyme A, we will get anyl coenzyme A, which gets changes into beta hydroxyacyl coenzyme A. This beta hydroxyacyl coenzyme A then further changes into beta ketoacyl coenzyme A, and at the end one acyl coenzyme A will be left and another 14 carbons will be there along with the chain. So this is a beta oxidation. This is your homework also. You have to draw this pathway. From palmitoyl coenzyme A, we will get inylyl coenzyme A. From it, we will get hydroxyacyl coenzyme A, and then beta ketoacyl coenzyme A. And at the end, acyl coenzyme A will be left, and the 16 carbon chain will get shifted to 14 carbon chain only. So that is all for this today's lecture. I will share the PPT with all of you, so that you will able to understand the things in detail. And that's all for today, because I think it's too much now. Yeah, you have to know about the disorders in. You have to know that which disorder is related to which disease only, and the diagnostic symptoms of that particular disease. Getting my point? So you have to know the diagnostic symptoms of that particular disease also. Otherwise, you have to keep it simple only. So I think that's all for today's lecture series. And with this, our whole syllabus is completed. Now, this is your homework also, which I already told you. So from the fact parts, you have to tell me about the beta oxidation of fatty acids. So is it clear to all of you? Please let me know. Please let me know. Is it clear to all of you? So that's all for today. Thank you all for your patience, and today is our last lecture day. So I wish you all the best for your future journey, for your future perspectives. And now we will meet only for the review paper. I will take the class maybe tomorrow on Google Meet or on MS Teams, so that we will discuss something about the review paper. Otherwise, everything related to theoretical part or this syllabus, we have completed everything. I will share the PPT with all of you so that you will able to discuss, or you will able to do it by your own self also. That's all from my side, and I wish all the best for to all of you for your future perspectives, for your future goals. And that is the thing which is there in the lecture series. If you want to learn more, something more from me, then I have my many classes which are running continuously, like genetics and PPT. So you can subscribe my channel, and you can get notification when I get live. And you all are welcome in my all lecture series. So yes, the homework which I told you is I also forget. I don't remember right now, but yes, you have to draw the metabolic pathways which are there in the carbohydrates, all the pathways in the carbohydrate metabolism. Number two, you have to let me know about the disorders which are there in the endocrine. Number three, you have to tell me the beta oxidation of fatty acids. So this is a homework for 2 days. So you can do it for 2 days. You can take 2 days for completing these three questions. So should I repeat again? The metabolism means the life cycles which are there in the carbohydrates, all the life cycles which are there, the biochemical cycles which are there. Number two, you have to tell me the disorders which are there in the endocrine, the hormonal disorders. Number two. Number three is you have to tell me about the beta oxidation of fatty acids. So that's all from my side. Thank you all. Thank you all for your patience, for being here with me everywhere. And I miss you a lot. I miss all of you for this biochemistry lecture series. But that is journey. We have to keep going. We all have to. So that is all from my, and from tomorrow onwards we will have no class. We will interact only on MS Teams. We will try. I will try to interact with all of you. I want that for these 13 or 14 days, I am the only one who speaks. Now I just want to listen your voice also. I want to listen and I want to see all of you. So I will try to take the class on MS Teams tomorrow, and it will be basically on the review paper part only. Get it? Getting my point? So the main thing is that the question will be basically review paper part only. Otherwise the lecture classes are over now, and we all are free from outside. And you have 2 days for submitting your assignments. But if you want to remain in touch with me, you are always welcome to be there in my another lecture series which will continue right now. So you can be there always, and you have to message me only. So you all are my students, so just try to keep in touch with me. That's all from my side. Please take care of yourself and keep growing, keep shining. My voice is not clear. Okay. I don't know what I speak now. I'm just saying, I'm just saying to all of you that keep growing, keep shining. Actually, what happening? I am just speaking continuously from 11 a.m. So that is why at that time my voice is very low, because my my voice is not now allowing me to speak, because I am continuously speaking from 11 a.m. and the sessions are very long today. I have concluded my 3 lecture series. That is why now my volume is little bit low. But I am just asking all of you that you all are welcome in my another lecture series which are there. You can be there at any time. If you want to listen something, you are most welcome. And even I will tomorrow I will have a lecture with all of you regarding the review paper only. So I will try to I will try to interact with all of you. I want to see all of you. I want to listen the voice of all of you. So tomorrow maybe I will try to take it on MS Teams, and so that I will like in this lecture series I am the only one who is speaking continuously. At so tomorrow I want the voice. I want that you all will speak. You all will speak, and I will listen only. So that is the thing, because you all are my students. You are very intelligent and you are very hardworking also. You are there always with me. Even the schedule, the timing is always not continuous. Sometimes we meet very early, sometimes we are in the late also. But still you are here along with me. So I want to thank all of you for your support, for your motivation which you gives me, that matters me a lot. And yes, this is the end of this lecture series, but we will meet tomorrow only for the review paper and only for the interaction round, so that to say goodbye to each other, that is the only thing. So I will share the timing and I will share the link of MS Teams also with all of you. The timings may be around 6 to 7 p.m. so that all will be free and they will join the session for a little bit, so that I will guide you regarding the review paper. So that's all from my side. If you want to say something, you are welcome. So that's all for today's session, and try to go through the PPTs. If you are not able to get anything, if any topic is not clear to you, no worries, we will discuss those topics again. Okay, don't worry. If something is not clear to you, if some topics are not clear to you, we will discuss it again. There is no problem in it. So that's all from my side. And thank you all. Thank you, Afreen, for your concern and for your appreciation. Thanks a lot, Afreen. And I want to say thank to all of you, and keep shining, keep grow. Yes, now I am free. From now I am free. So I by this night I will try to check the text from all of you. So we will meet tomorrow, but not for the lecture series, only for the review part. So till then, take care of yourself, and a very good night to all of you. Keep shining, keep growing. Okay, okay. So bye bye to all from my side, and I just not suggest, but I request to all of you to please remain stay in touch. We will definitely meet for another lecture series, definitely. So till then, take care of yourself. Good night. Bye bye. Take care. That's all from my side. We will meet in another lecture series, and we will meet tomorrow also for the review paper. Now I have to check your review papers, your abstract. So bye bye from my side. We will meet tomorrow. Bye bye.